Welcome everyone to our OpenSim webinar. My name is Jen Hicks. I'm the Associate Director of the National Center for Simulation and Rehab Research, and I'll be serving as the moderator for today's webinar. I'm pleased to welcome today's presenter. Jenny Yang is joining me here at Stanford. She will be presenting on tips and tricks for data collection, scaling, and inverse kinematics in OpenSim. Before we get started, a little bit about OpenSim in case you're new to the project. OpenSim is a freely available software application for visualizing musculoskeletal structures and simulating movements of humans and animals. So the first goal of our webinar series is to showcase the variety of cutting-edge research that's being performed with OpenSim. OpenSim is also a large and geographically diverse community of users, so the second goal of our webinar series is to provide an easy platform for the OpenSim community to communicate and collaborate and share some of the best practices and tips like Jenny's going to do today. A couple reminders about the format of the webinar. We definitely want to leave time at the end for questions and discussion, but we'll take those at the end of the webinar. And you'll enter those via the WebEx Q&A panel. If you need any technical help, you can consult the guide on our website or send the panelist or moderator a message on the WebEx interface. So now I'd like to go ahead and introduce our speaker. So Jenny is a graduate student in the Neuromuscular Biomechanics Lab at Stanford. She's the winner of both a Graduate Engineering Fellowship and BioX Fellowship from Stanford to support her PhD work. And she's an expert on measuring and analyzing human motion during sports and other tasks. Some of her work includes using OpenSim to study the differences between runners who use a rear foot and versus forefoot strike pattern, for example. Jenny's also a veteran staff member at our OpenSim Advanced User Workshops. So with that, really excited for your talk today, Jenny. Go ahead and take it away.
Thanks for the introduction, Jen. Hi, my name is Jenny Yang, and I'm a graduate student in Scott Delp's lab at Stanford. My current research focuses on studying the differences between rear foot and forefoot striking during running, as Jen said. And my project included collecting data in the lab and then using that data to create simulations of running. So the goal of today's webinar is to share our lab's best practices for scaling models and performing inverse kinematics. Getting accurate results for kinematics is important. Whether you're calculating joint angles to assess injury risk, like detecting high hip adduction angles in runners to assess the risk for tibial stress fractures, or using results to study muscle tendon lengths, which clinicians may use to determine whether someone with an impaired gait pattern is a good surgical candidate, or even just using kinematics results as a step toward creating accurate simulations. In order to have confidence in your kinematics results, you want to collect good data, accurately scale your model, and evaluate your results. Today's agenda will include discussing how constrained OpenSim models are beneficial compared to unconstrained marker-based methods for calculating kinematics, our tips on scaling models, registering markers, and kinematics tracking, and then how this information is useful for designing experiments. During this webinar, we'll explain our best practices and why we believe they improve confidence in our methods, as well as tips to avoid common mistakes. We're going to start by talking about OpenSim models compared to unconstrained models for calculating inverse kinematics. Before we begin our discussion on scaling or inverse kinematics, I'm going to start by talking about what we need in order to run these analyses in OpenSim. OpenSim models are comprised of several complex components, but for calculating kinematics, we require a model that includes bodies, such as the segments of the arm, the torso, and the leg, joints, which are used to connect bodies, such as this knee joint, which we use to connect the femur and the tibia, and model markers, which we track in order to calculate our hip, knee, and ankle angles. What separates OpenSim from other common marker-based methods for calculating kinematics is that our model is independent of the marker set. The primary benefit of having a model that's independent of the marker set is that the same model can be used with different marker sets in order to study different movement patterns. As I mentioned, we need marker sets to solve for kinematics, so I'm just going to discuss a couple right off the bat. So we start with our experimental markers, which are the data that we collect in the lab, and we compare them to model markers in order to track our subject's movements. Motion capture experiments typically involve collecting three-dimensional marker positions, as shown here by the blue dots. The goal of calculating kinematics is to take these experimental markers and use them to calculate joint angles, where joint angles are calculated by comparing the orientation of adjacent bodies. Shown here, we have reference frames on the pelvis, femur, and tibia, which would allow us to calculate hip and knee kinematics. With marker-based methods, we would use our markers to define the different reference frames, and then transformation matrices are used to relate the reference frames and calculate joint angles. However, in OpenSim, this process is different due to the underlying model. Instead, in each frame, we're trying to identify the joint angles that are necessary in order to minimize the error between the experimental markers, again shown in blue, and the model markers that are rigidly attached to each model body, shown in pink. Common marker-based methods use an unconstrained model that is defined purely based on marker positions. With these unconstrained models, you use markers to generate a bunch of bodies or reference frames, such as what you see in this figure to your right, and these bodies can all be considered independent of one another. What this means is that adjacent bodies can both rotate and translate with respect to one another. Because of how these bodies are defined, such that we are only interested in position and orientation of each body, one benefit of these methods is that we do not need to scale a model. Another benefit of using an unconstrained model completely defined by marker positions is that your model markers and experimental markers will align perfectly by definition. However, one consequence of having independent bodies that can translate with respect to adjacent bodies is that limb segments may appear to change length, since joint centers are not fixed with respect to the bodies. The primary difference between solving for kinematics using OpenSim instead of other common marker-based methods is that OpenSim uses a constrained model that is independent of the marker set. In OpenSim, our model is comprised of rigid bodies with joint centers at fixed locations in the body. As opposed to the unconstrained model in this figure, our bodies are constrained by the joints that connect them. Although our model markers won't perfectly match experimental markers, the result of how our bodies and joints are defined in the model is that the bodies will not translate with respect to adjacent bodies, and we don't have to worry about fluctuating segment lengths. Similar to how models in OpenSim constrain translational movement, the models in OpenSim use physiologically defined constraints on the rotational joints connecting adjacent bodies, which limit our rotations to realistic movements. In order to better visualize the issues that can arise from unconstrained models, here we have a model with no constraints, which is typical of marker-based methods. The femur moves freely, perfectly tracking the femur markers, but you can see the joint center move as the femur penetrates and dislocates from the pelvis. Using alternative modeling methods like plug-in gate or cast, it's challenging to detect these kinds of errors since the variables of interest are joint angles and not translational motion between the bodies. Using a model with joint constraints is important for musculoskeletal modeling, and it is because of modeling methods like consistent segment lengths and realistic joint motion that OpenSim models are appropriate for musculoskeletal modeling. In particular, it's important when trying to study muscles and tendon properties during movement. So in this figure, we've plotted the rectus femoris muscle tendon length during the gait cycle that you just saw. The unconstrained models-based methods are shown in red, while a model that includes joint constraints is shown in blue. Without joint constraints, we see that the muscle tendon length has a higher amplitude and more noise right here at the peaks. We believe that models with joint constraints provide more realistic values for muscle and tendon properties, and that's the primary reason that we use constrained models for calculating kinematics. Although scaling a model before running inverse kinematics can be a lot of work, the ability to accurately calculate muscle tendon properties and generate valid simulations of movement are why it's worth it. Based on that last example, hopefully you understand why we should use models with joint constraints for kinematic analysis, especially when hoping to study muscle and tendon properties. However, to use these models properly, they need to match the person from which the experimental data was collected. Although our model isn't defined by markers, we use markers to help us scale the model such that the size of the model is consistent with the person you're hoping to simulate and to orient the different limb segments in space. Model scaling can be tricky, and we're hoping to give you some insight into how we approach this process. To summarize what we've discussed so far, OpenSim models include rigid bodies and joint constraints. This figure is an example of an OpenSim model, and in addition to showing the different rigid bodies, we also visualize the various joints that connected them, such as pelvis tilt, pelvis rotation, hip flexion, knee flexion. As I mentioned, the joints will have rotational constraints in addition to translational constraints. For example, we represent the motion of the tibia with respect to the femur using only 1 degree of freedom, knee flexion. Although marker-based methods for kinematics perfectly track markers, they allow translational motion between bodies, which is problematic and often undetected. The OpenSim Inverse Kinematics tool solves for joint angles differently by posing the model to minimize marker error and limits the motion of the bodies. And finally, models with constraints are more accurate for musculotendon analysis. So the next thing we're going to talk about is scaling models. Ideally, we would use MRI images of a participant to scale the geometry of bodies and to place our model markers, but in reality, we don't have this type of data. Instead, we typically use marker-based motion capture data of a static trial to both scale the model and register the markers. The OpenSim Scale tool was developed to use marker-based data to do the most basic functions, scaling a generic model to match experimental marker measurements, and register model markers to match their experimental position. Before I get into all the details of scaling, I'm going to go back and describe some additional marker sets that you're going to be hearing me discuss today. So far, we've already defined our experimental marker set, which you see here in blue again. Again, these markers are the markers whose data was collected in the lab. Now, the markers that are going to be most useful for scaling are the anatomical markers, shown here in red, which are a subset of the experimental markers, and they're the ones that were placed on bony landmarks. So these markers should have been placed carefully on the subject and will hopefully be consistently placed from subject to subject. The alternate subset of markers that were collected during data collection but are not anatomical markers are known as tracking markers, and here we've shown them in green. These markers do not have to be placed consistently between subjects and are most important for tracking motion during dynamic trials. Additionally, these are the markers that are most important during the marker registration step, which I'll get to after we talk about scaling. The next marker set consists of virtual markers, which I've shown in purple. Virtual markers are not experimentally collected markers, but are calculated from experimentally collected markers. We generate this additional set of markers using our anatomical markers after data collection to facilitate the scaling process. Some virtual markers, like hip, knee, or ankle joint centers, may be calculated from functional trials or from regression equations. Other virtual markers are just calculated from nearby anatomical markers to help with scaling. And then finally, we have our model markers, which we've already discussed, shown in pink. We should have a model marker for every anatomical, tracking, and virtual marker. Okay, so back to model scaling. Scaling is performed by using marker pairs or sets of marker pairs to compute a scale factor for each body of interest, then applying that factor to the geometry, joint locations, mass, and inertia. In order to scale, we start with experimental markers placed on the subject and calculated virtual markers. We compare the distances between marker pairs from the experimental data with marker pairs taken from our generic model. We can identify our scale factors and scale our model by computing a ratio between experimental marker distances and marker distances on the model in order to get this scaled model, which I'm shown on the right. For example, we can see that the distance between the blue hip and knee joint center markers is larger than the distance between these same markers, shown in pink, on the generic model. So in this case, the scale factor for our femur would be greater than 1. Something else to note is that we scaled our femur using joint center markers, which are virtual markers. These aren't actual experimental markers that can be collected in a lab setting, but we compute them based on other experimental markers. Now, how you compute these virtual markers is up to you, but we strongly recommend using many virtual markers to assist you in scaling your model. In OpenSim, each rigid body is a three-dimensional object, with each axis being treated somewhat independently. Often we'll see people trying to scale all three axes of a body using one single scale factor. And while this method can work reasonably well for bodies that are mostly defined by one axis, we recommend against this. This is especially true for bodies like the pelvis, which don't necessarily scale uniformly across all dimensions. We're going to discuss our best practices for scaling the four main bodies of the lower limb, but beforehand, we want to start with our simpler example. So we're going to start by explaining how we would scale a rectangular prism. When deciding how many scale factors you want to use to scale a rigid body, whether you're using 1, 2, or 3 along each of the axes, you want to think about how much information you have along each axis, and you want to use marker pairs that are on or parallel to the axis of interest on the body. For example, with the pelvis, it's not hard to get information along all three axes, but for the femur, we rarely apply markers that will help us define a scale factor along the x-axis, or the axis that points from the back to the front of the knee. But back to our example. Let's say we start with the image shown with 3 experimentally collected marker positions. Now, we can add to this figure, we can add to this marker set by assuming we can calculate 2 additional virtual markers using some kind of regression equation or functional trial. However, note that we still don't have marker pairs aligned or parallel to all of our axes. So now we're going to add even more virtual markers to help us get marker pairs along all 3 axes. We're going to start with the x-axis that points from the back of the rectangular prism to the front. Now, looking at this cube in the middle, we still don't have any markers that are parallel to this red x-axis. However, if we create a virtual marker halfway between the 2 markers on the top, we can use this new virtual marker and the marker on the far side of the cube to define our axis. Now, using this cube in the center, you'll notice that we also don't have 2 markers that are parallel with the y-axis. So we're going to create 2 more virtual markers to help us calculate that scale factor. We're going to start by adding a marker at the halfway point between our last virtual marker and the marker at the far end of the cube. We're going to add another virtual marker at the halfway point between the 2 markers at the bottom of the cube. And now we can define a scale factor for our y-axis. The easiest scale factor to calculate is the z-axis. In this case, we're just going to use the 2 markers placed on the bottom. With the addition of these virtual markers, we can now define our model in all 3 dimensions. Now that we've shown how to generate these scale factors for a generic body in 3 dimensions, we're going to define the 3 axes of the pelvis in a similar way. So our typical marker set includes 4 pelvis markers, the right and left ACES markers, and the right and left PCIS markers. And we're first going to add the hip joint center markers. As I mentioned, these markers are usually calculated using regression equations or from functional movement trials. Since we often calculate these markers from functional trials, we have the most confidence in the placement of these markers. Next, we're going to add some markers to help represent the midline of the pelvis. So we're going to add a marker between the left and right ACES markers, which we'll call the mid-ACES marker right here. We're similarly going to pick a marker that's halfway between the right and left PCIS markers, and that's this one right here. And we're going to create a marker halfway between the right and left hip joint centers, which we'll call the mid-hip joint center marker right here. Finally, we're going to calculate a marker that's halfway between our mid-ACES and mid-PCIS markers, and we're going to call that marker the mid-pelvis marker. Now that we've added in our virtual markers, we're ready to choose our marker pairs that we're going to use for our scale factors. So for the x dimension, we recommend using the distance between the mid-PCIS and the mid-ACES markers. For the y dimension, which represents the height of the pelvis, we recommend using the distance between the mid-hip joint center marker and the mid-pelvis marker. And for the z dimension, we're going to recommend using the distance between the right and left hip joint center markers, since we have the most confidence in those marker positions. Next, we'll discuss scale factors for the femur and the tibia. I mentioned that our scale factors are limited by how much information we have for each body, and these are 2 bodies that we recommend scaling with 2 rather than 3 scale factors. We're going to have 1 scale factor that represents the length of these bodies, and then we'll assume rotational symmetry and scale the x and z axes using the same scale factor. The virtual marker that we add for the femur is the hip and knee joint center. And for the tibia, it's the knee joint center and the ankle joint center. For both the knee and ankle joint centers, you can calculate them using functional movements or just average the medial and lateral femoral condyle markers or the malleoli markers. Along the length of these bodies in the Y direction, we recommend using the distance between the hip joint center and knee joint center for the femur, and knee joint center and ankle joint center for the tibia. We suggest scaling the X and Z dimension using a scale factor calculated from marker pairs along the Z-axis. So for this scale factor, you'd use the distance between the medial and lateral markers placed on the joint. So for the femur, again, this is the medial and lateral femoral condyles. And for the tibia, you'd use markers placed on the medial and lateral malleoli. Now, depending on your subject, you can scale the left and right leg together, but we suggest scaling them separately. Using several virtual markers, our lab typically scales the foot in 3 dimensions. Note that when we scale the foot, we're scaling the talus, calcaneus, and toes all together. Also, our subjects most commonly wear shoes, and our goal in scaling the foot is to scale it not as a foot, but as a shoe. In order to scale the foot in 3 dimensions, we're going to need to incorporate a different type of virtual marker. Rather than using a marker calculated from functional movements or averaging between experimental markers, we're going to project markers placed on the shoe onto the ground. When applying markers to the foot during experiments, we will usually apply a marker on the heel of the shoe, which we place approximately level to a marker that we place on the 5th metatarsal, and finally a marker on the top of the shoe as close as possible to the 1st metatarsal, which we refer to as the toe marker. We tell our subjects to stand with their feet flat on the floor, but since we can still not guarantee that these markers are all on the same plane, we create virtual markers that project these 3 markers onto the ground and use these virtual markers to help us scale. In addition to our projected heel, toe, and 5th metatarsal marker, we also added an ankle joint center, a projected ankle joint center, and then a projected marker that's halfway between the projected 5th metatarsal and projected toe marker. In the X dimension, our scale factor is calculated using the distance between the projected heel marker and the projected toe marker. It's between the projected heel marker and the marker that's placed in between the projected toe and projected 5th metatarsal marker. In the Y dimension, we use the distance between the ankle joint center and the projected ankle joint center. And in the Z dimension, we use the distance between the projected toe marker and the projected 5th metatarsal marker. Now that you've generated your virtual markers, created your scale factors, and scaled the different bodies in your model, your instinct may be to ask, is my model scaling good enough? Unfortunately, there's still more work to be done before we can answer that question, but we are getting there eventually. To review what we've learned so far about the scaling tool, OpenSim scales the body of its models using scale factors which we calculate from marker pairs or sets of marker pairs. The best marker pairs for scale factors should be along or parallel to the axis of interest. Virtual markers, which are calculated with respect to anatomical markers, are very useful for scaling. The number of scale factors used for each body is dependent on how much information we can get from our marker set. As a reminder, we suggest 3 scale factors for the pelvis and the foot, and 2 scale factors for the femur and the tibia. We also decided that we're not ready to evaluate the accuracy of our model, but that's coming up. Based on what we've learned so far, we can also point out some common mistakes that we found with OpenSim scaling. So some of the most common errors we've seen include scaling all bodies uniformly or using only 1 scale factor per body, not using virtual markers to help improve scaling results by identifying marker pairs along the necessary axes, and scaling the left and right bodies together without considering whether or not it's appropriate. Before we can assess our scaling, the next step is marker registration. The scale tool in OpenSim does more than just scale the model. It is also helpful for marker registration or accurately placing tracking markers on the model. It's important to note that you should only be moving tracking markers and not any of the anatomical markers that are used for scaling. The reason we do not move markers used for scaling is that you should have placed these anatomical markers as accurately as possible, both on your subject and on your model beforehand, and you'll be using these markers to evaluate how well your model is scaled. Moving these markers could also potentially cause issues since these marker positions are needed to calculate scale factors. To minimize the chances of making this mistake, you can fix these markers in the marker setup file. Shown here is the OpenSim file for the model, which currently has an attached marker set. Now I've zoomed in on an important marker that I want to fix in the model, the right hip joint center marker. Before scaling and marker registration, you can change your anatomical and virtual markers to be fixed in the model, since we don't want these markers to move during the scaling or marker registration steps. Again, we use marker registration to move our tracking markers, or the markers that were placed less accurately on your subject. The tool will move those model markers so that they are perfectly aligned with experimental markers, and they will be fixed on the body to which they were previously assigned. So shown here, we're going to talk through an example of how marker registration works now. So we start with a scaled model with model markers. The first operation of marker placer is to basically run a short inverse kinematics analysis and to move the model into a position that matches the subject. To run this inverse kinematics analysis, we apply weightings to the markers based on how confident we are in their placement on the model. In this case, we heavily weighted our hip joint center markers since we are the most confident in their placement. We still weight our anatomical markers, but slightly less. And we provide no weighting for the thigh cluster of tracking markers. As a result, we see that the model markers for the joint centers line up really well, and the other anatomical markers also look pretty respectable. However, these tracking markers don't seem to match at all. So the next operation is to adjust the model markers so that they exactly match the positions of their experimental counterpart. There are two important things to note about what was done in this process. The first is that the inverse kinematics operation was dependent only on the joint center and anatomical markers. It's very important to have the model joint centers and the experimentally calculated joint centers be consistent, since these are likely to be our most accurate experimental markers. The second thing to note is that the cluster is left unweighted since we don't actually know their positions on the subject, and we want to ensure that they don't interfere with our initial model pose. Because of how this marker placer works, it's a very useful tool when markers move or fall off during an experiment. If you note which markers moved, even including anatomical markers, and collect another static trial, you can use the marker placer to accurately replace the model markers on your scaled model with respect to the markers that didn't move. When using a marker-based algorithm for calculating kinematics, where the markers define your rigid body, it is not possible to recreate the same model unless that marker is replaced in the exact same spot on your subject. After scaling your model and marker registration, we're now ready to answer the question if our model is good enough. The first thing to note is that simulation, specifically scaling a model, is a subjective process, which may initially make people uncomfortable. Two researchers, even from the same lab, might scale the model differently and come up with subtly different results. There's no one way to scale a model, and it's very possible you'll have to iterate on marker weightings and choosing scale factors. And while the subjectivity can make researchers feel uncomfortable, with experience and a methodical workflow, the process gets easier. Despite being a subjective process, there are also methods that you can incorporate to make sure you're on the right path. The first thing is to check the model pose and make sure it looks realistic compared to the static trial you collected. Ideally, you could compare the model to a photo of the subject while they're in their static pose. Another thing to check is the anatomical scaling markers that you didn't move during the marker placer process. Now in this image shown on the right, you can see that all of my tracking markers perfectly match their experimental counterpart due to the marker placer. But we can examine the errors between the hip joint center markers, shown in purple, and our anatomical markers, such as the pelvis markers or knee markers, and make sure that those errors are within an acceptable tolerance. The best way to evaluate your scaled model, though, is to evaluate additional results. If later on in the simulation pipeline, you notice that your inverse kinematics results or inverse dynamics results or even computed muscle control results don't look right and nothing seems to be helping, it's likely an issue with your scaled model. Ideally, you'll have set up a pipeline to quickly generate new results after adjusting your model so that having to correct your model doesn't completely interrupt your progress. Just as a side note, it can realistically take 10 to 12 hours to scale a model to one subject. And how long it takes will be dependent on the data collected, dependent on the marker set used, and dependent on the model chosen. The most important things to remember about marker registration include the following. Marker registration is also part of the scale tool and is primarily used to place tracking markers on the model. It is also useful for moving model markers when experimental markers may have moved or were replaced during data collection. Now, it's our recommendation to collect a new static trial after reapplying markers and noting which markers moved so that you can just move the model markers to match the new positioning of the experimental marker. However, if you forget to collect a new static trial, as long as you note the problematic markers, you can always just reduce the weighting of these markers during inverse kinematics. We also learned not to let anatomical markers move during the marker registration process. These markers are important for evaluating your scaled model after the scale tool. However, the best way to evaluate your scaled model is to continue assessing your results as you complete subsequent analysis. Based on what we learned about marker registration, I also want to quickly review some common mistakes when using this tool. When using marker registration, you don't want to weight tracking markers, and you don't want to move anatomical or virtual markers. When weighting markers, we have the most confidence in functional joint centers and a good amount of confidence in our anatomical markers. It's also a mistake to not take into account markers that move or were replaced during data collection. Finally, when scaling a model, you can anticipate that it will take several iterations. You want to be prepared for having to change your model at any step during your workflow and having to rerun all of your analyses. Now, we've briefly touched on how inverse kinematics works, but now we're going to talk more about setting up this tool and evaluating the results. On this slide, I've included the setup screen for inverse kinematics in OpenSim. Note that we require a scaled model with model markers and experimental markers for tracking our subject's movement. And using the weighting tab, we also require weights for markers that emphasize the markers that are most important for tracking. Similar to scaling, you want to weight the markers that you trust the highest and give smaller weights to markers which you have less confidence. For example, here we've highlighted the knee joint centers, and we can see that we've weighted them 20. Now compare that to the tracking markers on the left thigh, which are only given a weight of 1. Something to note about the weightings is that they're both used during the scale and inverse kinematic steps, and that something to note about the weightings that are used during the scale and inverse kinematic steps is that they are used proportionally, as in weighting the tracking markers with a 1, while weighting the joint center markers with a 20 would be the exact same as weighting the tracking markers with a 10 and the joint center markers with a 200. We generally suggest that weightings fall between 0 if you have no confidence in a marker's position and 20 when you are very confident, and never exceed 100. One benefit of having a model that uses weight to minimize errors across all markers is that it's resistant to individual markers with large errors, since you can always weight markers less or give them no weight at all. If you're worried about having noisy markers, having more tracking markers will give you more freedom in marker weighting and are therefore useful for calculating accurate joint angles. Shown here is the same setup file, but viewed outside of OpenSim as an XML file. Whether or not you're running inverse kinematics through OpenSim or through scripting, I want to direct you to one line of the setup file that's useful for evaluating your results. I've zoomed in on the tag for reporting model marker locations in ground. By setting this to true and using some simple scripting procedures, I can visually compare model marker positions and experimental marker positions in order to evaluate my results. When doing so, make sure you know which marker errors you are most interested in minimizing and which matter less. When it comes to asking if your model is good enough after running inverse kinematics, I should warn you that it's still subjective. However, similar to scaling, there are still results you can use to help you decide whether it's worth retuning or accepting your results. The easiest and most basic first step is to assess the inverse kinematics results visually by comparing your model markers to experimental markers. You can slowly run your model through the motion and visually inspect how well the marker sets match in each frame. By using that tag in the XML file and some simple scripting, you can also plot the errors between your markers. From top to bottom, shown here, are marker errors for the knee marker, the heel marker, and the toe marker as a percent of the gait cycle. While I can't give you an exact number that lets you know if kinematics results are good enough, in my study, I was able to get marker errors for my knee and for my heel to be around or below 2 centimeters. However, note that the errors for my toe markers are much higher and hit as high as 4 centimeters. When assessing your marker errors, it's important to know which markers you care the most about and which markers you may care less about, and most importantly, which markers will have larger errors due to the constraints on your model. So in the case of my simulations, in mid-swing, right here between 60 and 80% of the gait cycle, this subject had increased foot progression angle, which is a degree of freedom that's not included in our model. As a result, I allowed my toe marker to have greater errors while still being satisfied with my simulation results. Finally, another very straightforward way to evaluate your results are to plot your kinematics results. Shown here are some kinematics results for one of my subjects, and from top to bottom, I've plotted this subject's hip flexion, knee flexion, and ankle dorsiflexion angles as a function of percent gait cycle for both the left and right leg. The first check is comparing that the legs are pretty symmetrical, although this may not always be true depending on the motion that you're representing. Additionally, you can always compare your results to literature and ensure that they match the values that you expect. Note that good kinematics come from a well-scaled model. Bad results are typically not from poor weightings during the inverse kinematics setup, but from bad scaling. Okay, we ran through inverse kinematics pretty quickly, but the most important points are the following. Make sure that while weighting markers, you give the highest weightings to the markers that you trust the most. And if you notice that a marker has particularly noisy data, you can reduce or eliminate the weight of this marker. Because of the ability to change the weight of the markers, inverse kinematics benefits from having redundant tracking markers. Having additional tracking markers gives you more options when weighting markers and also reduces the importance of any given marker. When evaluating your results from inverse kinematics, you want to compare your kinematics to the literature and make sure they match the values you expect. If you expect symmetric motion, you can also compare the left and right limbs. Additionally, you can evaluate marker errors if you print model marker positions and write up a simple script to compare model markers to experimental markers. After reviewing several trials for several subjects, it will be easier for you to identify an acceptable range of marker errors. However, when reviewing marker errors, make sure you recognize which markers you care the most about and which markers may naturally have high errors since certain motions may not be included in the model. Finally, good kinematics come from good data and a well-scaled model. Now that we've talked through how scaling, marker registration, and inverse kinematics works, let's briefly discuss how we can design our experiments to facilitate generating valid simulations. The first step is making sure you're choosing a good marker set for the motions that you're trying to simulate. You should include any anatomical markers that you'd like to include for scaling purposes. For example, if you're planning on scaling the pelvis using the methods we suggested, you'll want right and left ACIS markers and right and left PCIS markers at a minimum. Make sure you place markers on bony landmarks as accurately as possible in your subjects. You want these markers to not only be consistent among subjects, but also ensure that model markers are placed with care in the same position on the model that the anatomical markers were placed on the subject. At a minimum, you need 3 markers to define position and orientation, and the same is true when modeling. For scaling, it's important to have these 3 markers be placed on bony landmarks that are fixed on each body. However, after scaling and marker registration, you can remove any anatomical markers that were used but make it in the way, as long as you have a sufficient number of tracking markers. Although having that minimum of 3 markers is less important since we're using a model with joint constraints, we strongly recommend having 3 markers, if not more, on each rigid body. As we've discussed earlier, inverse kinematics benefits from having many redundant markers, since you can put less weight on any given marker. When placing these tracking markers, we recommend spreading markers as far apart as possible to help minimize any local errors that may arise. This is most important on bodies with more fatty tissue like the thigh, and less important on bonier bodies like the tibia. As I mentioned, the marker set used for scaling may be different than the marker set used for tracking during dynamic movement. However, make sure you apply your tracking markers during your static trial, since you'll be placing them with respect to anatomical markers during marker registration. For static trials, you need the minimum of 3 markers placed on bony landmarks fixed on the body. However, some of these markers, particularly the ones that are likely to fall off during the motion, like medial knee markers or medial ankle markers, can be removed while tracking motion. Remember that although tracking markers are not necessarily placed consistently from subject to subject, the benefit of the marker registration step is that we have their position with respect to the body they are fixed to, and we can use them to track motion without anatomical markers placed on bony landmarks. Finally, make sure you have enough markers to model your motion of interest. If you're studying the foot and trying to represent multiple degrees of freedom, rather than treating it as a single rigid body, you'll want more than 3 markers on the foot. If you're interested in representing upper body movement, make sure you have enough markers on the upper body. The next step is discussing which data to collect. To start, you'll want to collect a static trial to use for scaling. Something we strongly recommend is taking pictures during the static trial, as these pictures will be very helpful during post-processing, such as when you're evaluating marker placement and your static pose. Pictures are also very useful when debugging, like trying to determine if your subject actually had different leg lengths or if they were just positioned in a weird way. You want to make sure that during your static trial, all markers are visible. You won't be able to scale or register markers properly if any of them are missing during the static trial. One more reminder that if markers fall off during the experiment, it's beneficial to collect another static trial and record which markers moved. If you know which markers were replaced on the subject, you can fix all of the other markers on the model while re-registering the position of the moved markers using the scale tool. If your model is defined by your markers, losing markers is a huge issue when trying to have consistent joint angles throughout an experiment, but it's less of an issue in OpenSim due to our model. You can always replace the markers using marker registration, but even if you forget to collect that extra static trial, as long as you know which markers moved, you can always just reduce or eliminate the weighting of those markers. Again, this demonstrates the usefulness of redundant markers. As I've mentioned, when we recommend calculating joint centers, specifically hip joint centers. When calculating joint centers, the goal is to calculate the positions of these points with respect to anatomical markers for which you have data. And this can be done using a regression equation relating the predicted hip joint center location to the anatomical pelvis markers. However, you can also relate hip joint centers to anatomical pelvis markers using an algorithm and dynamic movement trials. Note that you want to represent the range of motion in the movements that you care about. And you don't necessarily want the full range of motion, since you can get errors from skin stretch at the extremes. In our lab, we will typically have our subject move their legs in an asterisk-like pattern, like you've been looking at, and leg circles to calculate hip joint centers. Alternatively, you can use dynamic trials like walking or running to calculate your joint centers of the hip, knee, or ankle. When calculating data, you want to make sure that you're collecting high-quality data that is suitable for simulation. If you want more details on this subject, make sure to check out the page on collecting experimental data in the OpenSim documentation on Confluence. As a reminder, today we briefly talked about OpenSim models in the context of scaling and inverse kinematics, provided some tips on model scaling, marker registration, and kinematic tracking, and discussed our suggestions for how to design your data collection to facilitate the simulation process. The main points we hope you learned from this presentation include: constrained models are important for musculoskeletal modeling. Scale factors are generated from marker pairs or sets of marker pairs, and the number of scale factors used to scale a body vary based on information available to us from the marker set. We use virtual markers, calculated with respect to anatomical markers, to help us identify the most informative scale factors. Marker registration, another feature of the scale tool, is used to accurately place tracking markers. which is important when your method for calculating kinematics is minimizing overall marker error, but it should not be used to move anatomical or virtual markers. Inverse kinematics identifies the model pose that minimizes the error between model markers and experimental markers. And finally, good kinematics come from good data, which includes a proper marker set along with static and possibly functional movement trials and a well-scaled model. Hopefully this webinar provided you with some useful information to help you accurately scale your models, run inverse kinematics, and have confidence in your results. I would like to thank Chris Carty, Allison Arnold, and Apurva Rajagopal for providing content for this webinar. I'd also like to thank Jen Hicks, Joy Koo, and James Dunn for helping prepare the webinar, along with the rest of the Neuromuscular Biomechanics Lab for helping establish these methods for designing accurate simulations. And I'd also like to thank our funding sources. Now I'm happy to take questions.
All right, thank you, Jenny, for a great talk. That was super clear and lots of really great tips. Now we'll go ahead and get started with the Q&A session. As I said at the beginning, all the questions will be text-based. You can go to the Q&A box on the bottom right of your screen, type in your questions, and make sure you select to ask all panelists. So I think I've seen some questions already come in. Let's see. So first, a question from Dario Cazzola. First, he says, really useful tips. And his question, is there any way to use a combination of marker 3D positions and joint coordinates, i.e. joint angles from either direct kinematics or IMUs, to further improve the model scaling procedure?
That's a really great question, and it's definitely one that is useful, especially with the addition of more IMUs for helping with data collection. So the good news is that you can. So as I mentioned, part of the marker registration process includes a short inverse kinematics analysis where you pose the model based on your anatomical marker positions. Now, if you have other data from IMUs and you have more detail about your coordinates, you can actually also insert coordinate information during that process so that it's not just using your anatomical markers, but also making assumptions about your coordinates. You constrain the coordinates, and then you can use that to help pose your model such that during marker registration, you can check and see how well do your anatomical markers line up, and as I mentioned, move all of your tracking markers.
Great. So now a question from Ale Zhu. Thank you for the presentation. What do you use as values for acceptable tolerance for scaling NIK? Or does this vary depending on the study?
This is such an important question, and we get asked this question all the time. They're definitely subjective values, and that's why one of the things that we recommend is when going through this process, which can be very frustrating, you run through several trials, you run through several subjects, and you get an idea of just how well can you minimize your marker errors. And then when you see how well you can do, you can then go back and try and make all of your subjects and all of your trials match that data. So like in the example I showed, I was able to get my knee marker and my heel marker below 2 centimeters. And that did require allowing some wiggle room with my toe marker where you saw that it got up to 4 centimeters. But that's very typical of like my study and my results. Depending on the data that you collected and the model that you used and the markers that you used, you may be able to get smaller errors, but your errors it's entirely possible that your minimum errors are going to be higher and that your error tolerance is going to be higher.
Okay, now a question from Maxime Bourget, and I'm going to try to interpret the question, and hopefully I'm interpreting it correctly. So do you suggest to weight markers only on the confidence of their experimental acquisition, because you may maybe trust some bodies more than others because that body has more markers on it? I think is the question.
That's a really great question. And I think that you have to consider both. Like I said, when you're weighting your markers, this is the subjective part, and it's the most frustrating part of creating your simulations. And some of it is how confidently did I place that marker. Another is how much do I trust that that marker is going to stay in place during my dynamic trial. So for example, you're going to expect a lot of skin motion on the markers placed on your thigh, whereas you might expect less skin motion and markers placed on your tibia. So you should definitely take everything into account. Take into account how much you trust your placement, take into account how much you trust that marker's position throughout your trial, and play around with these numbers because that's what you're going to need to do to be confident in your results.
Okay, great. So now a question from Swithin Razu. Do the degree of freedom constraints at the knee, for example, a hinge joint, instead of having 3 or more degrees of freedom, affect the inverse kinematics results, and how can you tell the differences, I think, between a constrained versus unconstrained approach?
Yeah, so our knee is technically not a hinge joint, it's a coupled joint that allows some translational in addition to rotational motion, but it's just 1 degree of freedom. And then because of the fact that it's constrained, you most definitely can see your results in different results in kinematics. So when you're looking at a marker-based method for calculating your knee joint angles, you're actually going to find that your knee has 3 degrees of rotational freedom in addition to potentially some translational motion. So when you're using these direct marker methods, you'll find not only knee flexion, but you're also going to find other knee motions that we don't model in OpenSim. Now, if you look at your OpenSim results, the knee flexion angles will probably look relatively close, but you don't have any way to identify what those extra degrees of freedom are in the OpenSim model because they're just not modeled.
Yeah, so now a question from Martin Hora. Is it possible to scale differently 2 parts of one segment, for example, to prolong the posterior part of the foot and independently of the anterior part of the foot?
Absolutely. When you want to do this, though, this is going to be dependent on your model. So if you're going to try and take the foot, and our foot is comprised of these different bodies, we just constrain them all together and scale them all together when we scale. But what you're going to do is you're going to treat those bodies as independent. You're going to give them their own scale factors, and then you can scale them in the same way that you'd scale the femur and the tibia differently. Now, the only important thing to note is that if you're going to try and do that, you want to make sure that you have enough experimental markers to have confidence in how you're going to scale that part differently. And now, if you're trying to scale something like the femur in multiple parts, that's going to require a little bit more effort because that's going to require actually modifying the model file. You're going to want to take the femur and you're going to break it up into 2 different bodies if you want to scale them differently. And that's a long process that also requires coming up with inertial properties for those 2 different parts of the femur and so on.
Okay, so now a question from Nathan Smith. On the IK weightings tab, it looked like there were non-zero weights assigned for markers that were not checked as enabled. Does the enabled box have the same function as setting fixed to true or false in the OSIM file?
That's a really good question. I'm pretty sure it does. I'm pretty sure that if a marker is not enabled, it is the same as assigning it a 0 weighting. And I'm pretty sure that the enabled takes priority.
Okay, great. Question from Rachel Newman. Is there a template for how plug-in gait markers can be converted into marker pairs for OpenSim full body model scaling?
That's a really good question, and it would be a really great way to compare those 2 methods. I don't know of one right now, but it's not hard to take your data from that plug-in gait model and move it to OpenSim. It's just going to require designing a new OpenSim marker set that is reflective of your experimental markers, and then you'll have to set up the pairings that you use, but plug-in gait doesn't use marker pairings, so there's no way to try and take the marker pairings they use and use it for us. The way that they do it is they calculate the bodies as independent. They're not looking for marker errors. They're not looking for scale factors, as I mentioned.
Okay, now a question from Adam Clancy. This is a great presentation. Can you, it's moving here. Right there. Wait, there we go. Can you suggest a criteria for weighting markers during a task such as running?
That's a really great question. As I've been saying, this process is very subjective, but we would use the same recommendations that I gave earlier, which is that for the markers that you're most confident in when weighting, you want to use a 20, and for the markers you have no confidence in, you want to use a 0. Maybe use something like 1 for tracking markers, but it's the same rules apply. We recommend going between 0 and 20 and no more than 100.
Okay. A question from Daphne Pinto. How do you rotate axes by, say, 90 degrees in OpenSim?
That's a good question. I think, so if you're looking to rotate your body in OpenSim, I'm not sure if you can do that in the GUI. I think that's something that has to happen during the processing step. For example, when I collected my data, my lab frame, the up axis was Z, while in OpenSim it's Y, and it's a post-processing step to apply the transformation matrix to get our reference frames consistent between our data collection and in OpenSim.
Right, so you typically do that with pre-processing in MATLAB.
Yep, in MATLAB, we typically will convert our reference frames and move origins, such as for force plate data.
Yeah, okay, now a question from Ching Sai. I was informed that C-Motion Visual 3D software can help generate files used for running an OpenSim simulation. Can you provide more details about this or comment on whether the use of this third-party software may simplify the process? And maybe I'll jump in and answer that.
That would be
I'm
great.
not sure if you're familiar. So I think there are some tools, and they may help simplify the process. I haven't used them, so I can't affirmatively endorse them one way or the other. I guess something to be careful of is that you read the documentation that C-Motion provides really carefully so that you know what's happening on the C-Motion side. And so you understand any scaling that's already been done to the model or any transformation of the markers. So I would really recommend you talk to them and look at their documentation because they're making all the pre-processing software, and so they'll know best how to then bring that data into OpenSim. Okay, so I think we're getting to the end of our hour for the webinar here, so we'll go ahead and close up the webinar. If there are any questions we didn't get to, we can post some of the Q&A online after the talk, or feel free to follow up with us by email. Let's go through the last few slides. So thanks again for all the really great questions, and thank you again, Jenny, for a great talk. I want to acknowledge our funding sources. So OpenSim and this webinar series are supported by several grants from the NIH, including an NIH grant that funds our NCSRR. You can learn more about the OpenSim community and find other resources at our website. And we ask that you please complete the survey that will appear in a pop-up window when you log out of the webinar. This will help us improve the webinar series and choose upcoming topics. So thank you again, and see everyone at the next webinar.